It's really to help, through these studies, to help you understand how your body works. Because in science, medical science, we don't study health. We study diseases. You know, if you take 50 healthy people, you give them a carrot every day, 6 months later they're still healthy, you can't say carrot are good for health. You need to start from a problem and change it to be able to draw conclusion, you know, about how the body works. So that's essentially what I'll be doing. So I'll show you a number of studies, but let's understand it's all about how your body works, and I'm using them to illustrate the concepts that I'm sharing with you, so that you understand that it's not just me saying it, but there's really a lot of science behind this. Okay, so when we talk about stem cells, the reason why so many scientists are interested about stem cells worldwide, it's because of the global recognition that stem cells have enormous regenerative potential. So we know that stem cells have the ability of repairing, regenerating tissues. So scientists will take stem cells from your bloodstream, from your bone marrow, from your fat tissue, umbilical cords, will tweak them or not in the lab, and then re-inject them into your bloodstream. But one thing that is never talked about, which to me was really the most, I would say challenging, but the most interesting question, is that if stem cells are in your blood to begin with, they are in your bone marrow, they are in your fat tissue, and they have such amazing regenerative potential, they're not better because you take them out and you put them back in. So they're already there. So if they have such potential, what are they doing in the body? And when we do understand what stem cells are doing in the body, it gives us a new understanding of health and wellness. And I believe that it's going to change the way that we practice medicine. Push, let's say, 10, 15 years down the road, we will have a different medicine once all of this knowledge is integrated into the full practice of medicine. So let's get into the topic. To begin with, what is a stem cell? The best way to understand what a stem cell is to first define what is not a stem cell. All the cells of your body are referred to as somatic cells. These are all the cells of your tissues, your muscle cells, your cells of your immune system, of hormonal glands, your brain. If you ask a pathologist to look at a cell, the pathologist will be able to identify every single cell based on its physical characteristics. These cells will never change, they will never multiply, they are specialized, they do one thing. If you go to the gym and you work really, really hard and you need more muscle cells, the skin is not going to look down and say, gee, the muscle needs assistance, I'll go and become a muscle cell. It won't happen. So they are fixed, they do not transform. At the other end of the spectrum, you have stem cells. Stem cells are nothing. What I mean by this is that a stem cell in your body, by its nature of being a stem cell, does not do anything. It does not accomplish any function. Its function is in its ability to transform into a somatic cell that then will do something in your body. So they are the precursor of all these cells. Now traditionally, when I say traditionally, I mean still today in med school, what med students are told is that stem cells are essentially precursors to blood cells. They transform into red blood cells, white blood cells, and platelets. The fact that they can become cells of other tissue, it's sort of an exception. It's sort of some sort of, I wouldn't say weird information, but it's not the main role of stem cells. What I want to share with you today is that, yes, they are precursors to blood cells, but another one of their key vital role in the body is that, yes, they do become other cells. It's a normal part of your physiology. They are your repair system. So let's look at this transformation into one study, because I want to illustrate what I'm telling you, and it will lead us to answering a very, I think, interesting question. Here's a study in which scientists took a double beaker, so a small beaker inserted into a bigger beaker, in which we have water here that is bathing both beaker, because at the bottom here I have a semi-permeable membrane. So it allows molecules to go through, but it prevents cells from going through. So I have at the top here stem cells, I have at the bottom a piece of damaged liver, but molecules from that damaged liver can go and talk to these cells. And with this setup, 8 hours later, the cells started to transform into liver cells. The reason I could show this phenomenon with hundreds of studies, but that specific studies gives us a very interesting piece of information. Why is it in our world today, when we can go to Mars, the stuff that we can do scientifically is unbelievable. And we discovered today that our stem cells is our repair system. Not only we discover it, but as you'll see as I keep going, it's obvious. There's so thousands of studies. Why didn't we see this before? And the reason is because if you use traditional science to identify these cells, a liver cell, you put it under the microscope, a pathologist will look at it according to its shape, physical characteristics, everything that is visible about a cell, and he will tell you, this is a liver cell, but he cannot tell you, yesterday it was a stem cell. The process is completely invisible. It's a little bit like the first time that you see, we've seen caterpillars all our life, we've seen butterflies all our life. The first time that you see a caterpillar transforming into a butterfly, it's like eureka. You discover a reality of nature. Same thing here. We've seen stem cells, we've seen cells of different parts, different cells in the body. The transformation has never been seen until the day that we developed a marker. What I mean by a marker is a very normal fact of life. You're well aware of this right now. You all know the GPS. How did we discover? We have seen whales in deep ocean. We've seen whales by the shore. We never thought that the whales by the shore were those who could swim immense distance until we tagged them with a GPS. And then we discovered the phenomenon. The GPS for a cell is green fluorescent protein. It's a protein extracted from a jellyfish that is spontaneously fluorescent. And because it's a protein, we can derive the DNA that codes for this protein. You can inject that DNA in the nucleus of a stem cell. That means that stem cells will make this protein, but all the cells that derive from this stem cell will also make that protein. So if they all become liver cells, you look at them under the microscope, you can tell that they are liver cell. You just use a bright light and they start to glow. Now you can tell these liver cells were stem cells the day before. So it basically suddenly reveals the phenomenon in a remarkable way. So here's the typical studies that were done by many, many scientific groups. You irradiate mice, which kills stem cells, inject fluorescent stem cells, trigger an injury to a tissue, and then you observe the recovery process. Here's a study that we did. So this is the muscle in the leg of a mouse, injected with cardiotoxin, so it essentially destroyed the muscle, the leg is paralyzed. 6 weeks later, the leg has full mobility, it is completely green, completely reconstructed from cells that were derived from the bone marrow. If you trigger a heart attack, same thing, the area of the heart is now filled with green cells. If this is in the skin, this is in somewhere, in the kidney, this is in the lung, and then the last one, I'll come back to that later, this is a cell of the pancreas, a normal cell of the pancreas making insulin that is derived from the bone marrow. So essentially, stem cells can become cells of virtually any tissue of the body. So let's go back to my caterpillar and butterfly. The first time that a biologist observes a caterpillar that transforms into a butterfly, you can reach two conclusions. You can say, that's a weird caterpillar. Or you can be a little bit more daring and think, nature is organized. If one caterpillar became a butterfly, I've got to really look at all the other butterflies. It looks like butterfly comes from caterpillars. So what we did with this information, we thought, well, if stem cells can become, the early studies published in 2001, stem cells were documented to become brain cells, liver cells, and heart cells. So we thought, if they can become heart cells, liver cells, and brain cells, why not pancreatic cells? Why not lung cells, skin cells, other type of cells? It would be weird to see stem cells becoming these three tissues and not the others. So it had to be the repair system of the body. So getting the science that existed at the time, we published a paper in a journal called Medical Hypotheses, and we proposed the fact that stem cells appear to be the natural repair system of the body. Let's fast forward now 18 years later, and here's what we can tell of how this whole process works in the body. This is now supported by hundreds, if not thousands of studies. This is no longer an idea. This is what is happening in your body. Anytime there is an injury of any tissue, any part of your body, a lot of these studies were done with heart attack, stroke, burn to the skin, broken bone. If you have stroke, did I mention? Yeah. If once you have an injury, within hours after the injury, stem cells will leave the tissue, go to the bone marrow, trigger the release of stem cells from the bone marrow, and as the stem cells leave the bone marrow matrix, they get into the blood circulation. Now, as they leave the bone marrow and start to circulate into the bloodstream, the number of stem cells in the blood will increase anywhere between 3 to 10-fold. So these stem cells now, how did it get there? Okay, we need to go back quite a bit. Can you go back to where it was? This is like, can you help me a little bit? I don't know why it skipped that quick. It skipped about 10 slides. Okay, there you go. So the number of stem cells increase in circulation. Now these stem cells obviously don't know where to go. So this is why sometimes after a heart attack, people hear voices. It's the stem cell saying, where do I go? No, it's not true. So these stem cells circulate everywhere. So how do they identify where is the problem? What happens is that on the third day, starting on the second day after the injury, the tissue will release specific compounds locally in the area. This button is not the best. Okay, will release locally specific compounds that will remain locally, but as stem cells circulate everywhere in the body, and they circulate into the fine blood vessels of the affected tissue, locally, as these molecules touch the membrane of the stem cells, it triggers the process of migration. So adhesion molecule will be expressed on the surface of the stem cells. They will grab the capillary. They will secrete enzyme that will digest the attachment between cells of the capillary. They will cross the capillary wall, keep seeing where the injury is. They will crawl to the site of the injury, and when they touch the tissue, they start to proliferate and become cells of that tissue. It's a beautiful process, very well documented. So think about this. You cut your finger, you have a bacteria, what happens? You will have immune cells released from lymph nodes. They will go everywhere in the body, but they will not go into your nose or your ear or your big toe. They'll go exactly where you have a bacteria. It is a very, very well orchestrated phenomenon that we call the immune system. For a cell of the size of a cell to travel into your entire body and go exactly beside the bacteria to kill it, and think that this is random, it will be like you walking out of the hotel here and thinking that you will randomly meet your mother in Tokyo. It's never going to happen. It needs to be organized. So that's the organization of your immune system. It's the same thing that we have here. It's the organization of your repair system. You call for stem cells and you lead them exactly where they need to go to repair the tissue. So in this whole process, you've got the release of stem cells from the bone marrow, their circulation, their migration into the tissue, where they proliferate and differentiate or transform into cells of that tissue. Of all these different steps, in science, I'm saying this because I'm assuming some people here are not scientists, just to understand how science works. You can only observe the things for which you have a tool to make the observation. You cannot study bacteria if all you have is a telescope. And you cannot study stars if all you have is a microscope. So you cannot study circulation, migration, or these parts if you don't have something to stimulate them or block them. We have molecules to trigger the release. It's these molecule release, SOS molecule released by an injured tissue. So we can take these, inject them into animals or people, put more stem cells in circulation, and then study the link between the number of stem cells in circulation and health. So I'm passing all the studies. Here's the main conclusion, and then I will illustrate them with studies. First big conclusion, more stem cells in circulation means that more stem cells are available to participate in the process of tissue repair. That means I get two people with the same problem, burned skin, broken bone, same thing. The one that has more stem cells will recover faster. It's that simple. Here's one study, just to illustrate this concept. Doctors took patients who had a heart attack, then separated them into a group. The people who recovered very quickly, resumed an active life, a productive life. The other ones who did not recover very well, stayed in the hospital or passed. You count the number of stem cells in the bloodstream of these people, and this is people with poor outcome and better outcome. Half the number of stem cells. And there are many other studies. I'm just showing you one illustration. Second conclusion, if more stem cells mean that you can repair better, then it begs the question, the one that is not repairing as fast, what if I put more stem cells in their bloodstream? I have stem cells in my bone marrow. We have shown that they can be released naturally. It's part of your repair process. What if I have a means of stimulating this and putting more stem cells in circulation? Can I accelerate the process of tissue repair? Here's a study on the heart. And by the way, this whole process here is referred to as endogenous stem cell mobilization, the release of your own stem cells. When I give these lectures to the medical community, that is what we talk about. We have stem cell injection, putting more stem cells in circulation through an injection, and now we're talking about putting more stem cells in circulation simply by supporting a natural process already taking place in your body. So let's look at the heart. This is a study done by the National Institute of Health in which scientists took mice in which they stopped the blood flow to the heart, so it triggers a heart attack. Then they let them recover. They separated in two groups, one control group to which they did nothing, and an experimental group in which they injected these compounds known to trigger stem cell release from the bone marrow, increasing the number of stem cells in that study quite dramatically, about 1,000 times. So, and then they looked at these animals 27 days later. In the control group, you see severe scar tissue in the heart, no new blood vessels, 17% survival, and poor cardiovascular health. In the experimental group, we have complete renewal of the ventricular wall, new blood vessels tapping in the other ventricles, 73% survival, and quasi-normalized cardiovascular function. So in a world where there's nothing we can do to recover from a heart attack, here's an 18-year-old study that is showing if you have more stem cells in circulation. As done in this study with mice, that same protocol cannot be used in humans. I will not go into the detail, but the study itself shows us something very, very important. If we have a way to put more stem cells in circulation, the body does have a way of using them for tissue repair. Here's another one touching the pancreas. To study whether this phenomenon can also help a condition of, let's say, diabetes, we need to show two things. We need to show that stem cells can naturally leave the bone marrow, go to the pancreas, and become insulin-producing cells in the pancreas normally. We need to show that this is a normal part of the body. If it is a normal part of the body, then the question is, against that normal phenomenon, can I reach some sort of clinical benefits? Good enough? First study, a brilliant study. Scientists took male mice in which they did a genetic manipulation. They coupled the gene of green fluorescent protein to the gene of insulin. So what they do, just to make it clear, the first cell, embryonic stem cells that will become that mouse, that cell, I go in there and I do this genetic manipulation. Then I let the animal develop. So that full mouse here, any cell in that body that starts to make insulin will also make the green fluorescent protein, so it will be green. So the pancreas of that mouse is green. Now I irradiate these female mice, which kills all the stem cells, and I inject stem cells from these male mice. So at the end, I've got a female mouse. Just to summarize all of this and make it simple, I have a female mouse. The entire body is made of cells that have double X, no Y chromosome. The stem cells in the bone marrow have been replaced by stem cells that come from a male, so they have a Y chromosome, so I can track them in the body. And if any one of them starts to make insulin, it will turn green, so I can see it. So they isolated cells from the pancreas a few weeks later, and if you look at that cell, it has transcribed the gene for insulin. Now the question is it making insulin? By using a red dye for insulin, that cell is indeed making insulin. And observe that it's making insulin right between these other two cells. It is not a Frankenstein cell, sluggish cells, overactive cells, weird cell. It's just a very normal cell of the pancreas. At the age that we all have here, we don't have one cell in our pancreas that we're born with. That's what happened to all of them. And it has the white cell, it does come from the bone marrow. So that study alone illustrates in a very simple way that the stem cells from your bone marrow can go to the pancreas and become insulin-producing cells in the pancreas. Now the next step is, if I can enhance this natural phenomenon, can I have an effect on diabetes? Now, in medical research, we use animal studies. I'm sure you've heard about it. What you may not have thought about, or thinking through the whole process, is that you cannot find a diabetic mouse. If you want to study Alzheimer's, you won't find a mouse that forgot what a cat is. You won't find a mouse that has Parkinson or whatever the problems. They're just mouse. So you need to find a way to induce in them something that resembles a human disease or is the identical of the human disease, and then try to do something about it. So for example, Alzheimer's, they make a hole in the skull, you put aluminum, and then within a few weeks, the rat will have the same type of disease as Alzheimer's is, as an example. For diabetes, there's a toxin called streptozotocin. So this is the normal island of Langerhans in the pancreas that makes insulin. This is what it looks like. If you inject streptozotocin, it's a bacterial toxin, this becomes this in 24 to 48 hours. These animals no longer make insulin, they are diabetic. They need insulin to survive. So what did it is that they injected the same toxin along with endogenous stem cell mobilization, releasing the animal's own stem cells. And you can see that you have a partial recovery of these island of Langerhans, but it's enough to maintain proper function of the pancreas. These animals are making enough insulin so that they're fine. They're not diabetic anymore. Similar studies were done in humans, 14 humans, recently diagnosed for insulin-dependent diabetes. They were given 2 rounds of stem cell release, their own stem cells, and of those 14, 13 were independent from insulin between 3 to 36 months. I'm not telling you we found a treatment for diabetes. I just want you to see the power of your own stem cells sitting in your bone marrow. You renew your pancreas every about 4 years. That means the reason why you're not diabetic today is because the stem cells did their job during your entire life. They do it, otherwise you would not be here. Okay. And the third conclusion is sort of the corollary of the reverse of the first two. The first two say, if I have more stem cells, or if I can put more stem cells, I can repair better. The contrary, or the flip side of this, is that if I don't have as many as normal, if I'm deficient in stem cells, then I'm more exposed to develop various kinds of problems. So let's talk more about this one, because to me, this is the most important part. If there's a take-home message, it is this message. And it comes from our general understanding of disease formation. We never talk about this way, but if you think about it, how do we understand disease formation? Disease formation is cellular loss. If you lose cells in the pancreas making insulin, you have diabetes. If you lose cells making dopamine in the brain, you have Parkinson. If you lose cells making acetylcholine, you have Alzheimer's. You lose cells making T3, T4, hypothyroidism. Think of any disease, any degenerative disease, it is always the loss of a type of cell or a kind of cellular function, right? And the general understanding is that we are born, we mature, we reach about 25 years of age, peak health, and then everything starts to decline. Isn't this sort of the experience that we have? That's how we understand health. So this is a little bit like telling you you're born with a certain bank account, that's your genetics, spend it wisely. Okay? And then as we advance in age and we lose cells, where we will lose more cells than other, this is where the tissue will start to show signs, and that will be your problem. Diabetes for one, Parkinson for another one. So we develop all kinds of different problems. But stem cell research is showing us something different. Stem cell research is revealing that health is actually a balance between two phenomena, light and darkness. No. Is it on my end? There you go. Two phenomena. Cellular loss and tissue renewal. I don't have the slide here because I didn't want to make it too complicated, but there are very interesting studies that have clearly demonstrated your body is in a constant process of tissue renewal. 5-10 years ago, the science was, and then before that, you were born, up to about 5-10 years of age, your heart matures, and that's it. This is your heart. Anything you lose from your heart cannot be replaced. But your brain is the same. Now it's very clear, you replace your heart constantly, you replace your brain constantly, you have half of a new heart during your lifetime, you have a new liver every 2-3 years, new lung every 4 or 5 years. The time here is not relevant. The concept is that there's a continual turnover. You lose cells every day, you replace them, so life is like, you've got a bank account, you've got a regular income, as long as it matches your expenses, you're fine. That's what it means. When you're young, you've got a lot of... I promise I didn't do anything. Okay. So you're young, you've got a lot of stem cells, so you repair very, very well. As you age, there's a point where, as we age, the red marrow transforms into yellow marrow, fatty marrow that does not make stem cells. So our stem cell number declines as we age. There's a point where we cross that line when cellular loss gains ground. That's when we start to develop problems. We've not developed them yet. It's like the day that your income no longer meets your expenses, you're not bankrupt yet. You're just stressed. You're thinking about it, and it's slowly happening. And 2-3 years down the road, it will show up. It's the same thing. You can go for years in this area of deficit, and you don't have a problem yet. You just know that your health is not as good as it could be. And what is the difference between somebody who will develop diabetes or Parkinson, or somebody needs a titanium knee? It's a whole slew of factors. It's your genetics, your past injuries, your exposure to environmental toxins, your lifestyle, your exposure to physical stress, emotional stress, your diet, your ability to rest, your mother-in-law. All of this is the pressure that is applied on you and your ability to deal with it. You're not learning anything, you all know all of this. But when it relates to stem cells, if I take back my financial analogy, it's like to say, we've got somebody who cannot pay for their house, somebody cannot pay for their car, somebody else cannot pay for their health, somebody else cannot pay for their education. So we have car problem, house problem, education problem, and health problems. Everybody in this room, we all have different problems. They're all different. There's no two problems the same. But we probably can resolve all of them with $1 million, wouldn't we? The common denominator is that you don't have enough money. The common denominator is that you don't have enough stem cells. If you had enough stem cells, you could tilt that balance, replace the cells that are being lost, and your body will continue. If that is true, there's one way to test it. I can count the number of stem cells in people who have developed various kinds of age-related problems and compare that with the number of stem cells in healthy people. We proposed this about 10 years ago. Now a lot of these studies have been done and published. So here's a few of them. This is the number of stem cells in the bloodstream of normal people, normal glucose tolerance, impaired fasting glucose, impaired glucose tolerance, and diabetes. You see a straight line. As people move forward in the problem, they have fewer and fewer stem cells. Here's kidney function. Normal people, people who have various kinds of kidney disease, poor kidney function. Not only do we have fewer stem cells, but this is as they advance in the process of kidney failure. Fewer and fewer stem cells. There's a direct link between how many stem cells in the blood and kidney function. Here's another one. People who complained of poor erectile function, controlled individuals, healthy individuals. If you bring the average on the axis here, a little bit less than half the number of stem cells in people who complained of poor erectile function. You take all these individuals and you establish their international index of erectile function, and you do a graph between number of stem cells and erectile function, straight line. There's a direct link between how many stem cells you have and erectile function. It's a popular slide. Okay? This erectile function has a lot to do with vascular health. Vascular health in stem cells is probably one of the most widely documented relationship when we talk about stem cells. The link between stem cells and vascular health. Here's one of those studies. The link between one problem that sort of summarizes vascular health, high blood pressure. People with high blood pressure have on average half the number of stem cells found in healthy people. Here's another one with heart disease. As people have reached full development of ischemic heart disease, they have half of the number of stem cells in circulation. I think I have another one, Crohn's disease, ulcerative colitis. COPD, lung problem. We get the same thing with arthritis, lupus, metabolic syndrome. Essentially, every study that has been, disease that has been studied, we see the same conclusion. So we see the same relationship. So the big conclusion here, now forget all the disease. Am I the first one that has a button problem? So I guess it's me. There you go. So now forget all the diseases, because we cannot show this phenomenon without tapping into diseases. Forget all the diseases. The main conclusion is simply to understand that more stem cells in circulation means that your body is better able to repair and better able to maintain itself day to day. And to me, where the big conclusion is for this in terms of society, when I say it's called to change the way that we practice medicine, is that today the best of medicine is preventive medicine, is it not? But if you think about it, it's like saying the best way to be wealthy is to prevent poverty. Will you ever be wealthy if your strategy is to prevent poverty? You'll just be not poor. If you want to be wealthy, you need a strategy for wealth. So here we are, trying to achieve optimal health, while the aim is just to prevent the problem. But it was the best that we could do, because we did not know that the body had stem cells before. Once we discovered that the body has an innate, intrinsic system that is aimed at keeping us healthy, then it opens the door to a whole new way of looking at health, which is we can now enhance the ability of the body to have its own optimal health. If you think about it for a second, how do we define health in our society? The absence of disease. Go tell your boss and tell him, I feel in 2-3 weeks I will have a heart attack. I need 2 weeks in the Seychelles. You won't get it. You have your heart attack, you will have 6 months, you're fine. We think of we used to climb stairs 3 by 3. 5 years ago, 2 by 2. 2 years ago, 1 by 1. 6 months ago, 1 by 1 slowly. Then we stop in the middle to catch our breath. We have a heart attack. Then you're a heart patient. Before that, you would just, okay, life is just going on. You're not performing at all, but you're not sick. You don't have a disease. But it's clear that the diagnostic is an illusion in the whole process. Everything is a slow evolution until our society has decided to establish a criteria that now we call you sick. Everything is a gradient. It's a gradient, and it evolves because we lose cells and we don't replace them with the same rate as we lose them. If you can put more stem cells in circulation, you just give back that edge to the body to just maintain itself. It's really all it is. It's a very, very small thing, but it can be huge for your body. So how can we do this? That's where comes Revitably. So all of this started when I was studying. So we go back to 1994. I was studying a blue-green algae growing in Klamath Lake in southern Oregon. I was hired by a company to study how that plant was working. People were reporting a broad variety of health benefits, and we were not able to explain how one plant could have benefits on the heart, on the skin, on joints, on the lung, on the pancreas, on the liver. We had no explanation for a number of years, until the day that scientists started to document that stem cells could become heart cells, liver cells, and brain cells. We thought, what if they can become also pancreatic cells and skin cells? What if they were the repair system? And if they are, what if that plant works by stimulating their release and we put more in the blood circulation? At the time, 2 very far-fetched ideas. The number of times that I was told I was an idiot, it was many times, but it turned out to be what it is. So we developed an extract, we tested it, and after about an hour after consumption, we get a significant increase in the number of circulating stem cells. That's the main mechanism of action of this product. Once we discovered this, it was clear to me that AFA cannot be the only plant in nature that has an effect on stem cells. So we started to ask questions like, what other plants have been known historically to be associated to a wide variety of health benefits, or things like longevity? What comes to mind right away is things like goji berry, medicinal mushroom, fucoida. We tested all of them. We saw that they all have, to some degree, an effect on stem cells. And then we started to look locally in various areas of the world, South America, Papua New Guinea, Madagascar. Are there plants that they're using locally for a broad variety of health benefits? And the latest one that I studied that was really interesting is sea buckthorn berry. coming from the Tibetan plateau. So Sibok thorn, its scientific name is Ipophyte. So this is the field where we filmed this shot. So this is the field where it's coming from. So it's the Tibetan plateau into national parks. And the name for Sibok thorn is Ipophyte. It's very interesting because in science, we normally name plants according to their physical characteristics. For example, the plant before is Aphanizomenon flos aqua, which means invisible flower of water. It looks like a nice plant in water. This one is Ipophyte. It's horse light. It means shining horse. It's Alexander the Great traveling into Western China in these areas during his conquest, and at some point he abandoned a group of wounded horses, wounded in battle, and he just left them to die in a field of Sibok thorn. Came back a few weeks later, they were healthy, vibrant, with a shiny coat. A shiny coat for a horse means health. A healthy horse has a shiny coat. So the berry was named Shining Horse. From there it was integrated into Tibetan medicine, Chinese medicine, Mongolian medicine, where it has been used. And if we go to the next clip is the harvesters that are harvesting in the field where we take the berries. So the main use historically has been lung problems, lung health, heart health, cardiovascular health, good blood sugar, pancreatic health, and also injuries, burn to the skin, cut to the skin, broken bones. So if you think about lung, heart, cardiovascular, skin injuries, to me it means stem cells. So we developed an extract of it, we tested it, and we found a very interesting benefit. Compared to AFA that was a transient release, here we got a more sustained increase in the number of circulating stem cells. We have the same thing with aloe. The aloe that we have is a very, very unique aloe. We discovered this after going to Madagascar and looking at various plants, and at some point, it's a friend of mine, biochemist, who went there to look at plants for Parkinson and Alzheimer's. And as he's leaving, his guide is telling him, you've got to study this plant. This is the best that we have in Madagascar. He said, study this. Obviously, he didn't know what to study it for, so he kept it in his fridge for 5 years until I shared with him what I was looking for, and he told me, well, look at this, what it does with stem cells. And we saw a very, very interesting reaction. We tested many aloes, and then we found only one other aloe extract that had this effect, which is what we have in Revita Blue. So we see a significant increase in the number of circulating stem cells. So Revita Blue is essentially a blend of the Sibok thorn extract in a full dose as documented in the papers that we published in the scientific literature, AFA, aloe vera extract, and beta-glucan that has also been documented historically to support stem cell function in the body. That's what Revita Blue is. Now, when I started to develop Revita Blue, Dr. Entar, she's on the scientific advisory board, told me, you've got to go and study the product in Jeunesse product line because you'll be surprised to see how Revita Blue complements a lot of the product that are already existing. So let's go through them a little bit because it is totally true. So as we release stem cells with Revita Blue, there's something in the body that can suppress the ability of stem cells to really see the tissue that is in need of repair. And it is systemic inflammation or oxidative stress, free radicals in the blood. So if you can have a strong antioxidant to reduce free radicals, you will clear the noise and allow stem cells to better see what they can go. So you have that with Reserve. So both Revita Blue and Reserve will have their own benefits. But as you take them together, the cells that you have, the stem cells you have released will be more effective to find their way to a tissue or an organ. So another interesting synergy is with Luminesse. So I briefly talked about this morning, how the skin really renews itself. So it's with stem cells coming from the bone marrow to the blood to the skin, and then you renew the skin. Well, if you put more stem cells in circulation with Revita Blue, it will already help the skin. It's not rare for people to just take Revita Blue and people will tell them, or they come back and they say, people are telling me after a month or two, what are you doing? You look more radiant. Stem cells will go to the skin. Now apply Luminesse that has peptides that will attract these stem cells from the blood and bring them to the skin. So you really enhance now the effect of the Revita Blue. Both of them will work together very well. As you release stem cells, one stem cell can become anywhere between 1,000 to 5,000 tissue cells. So as you release, let's say 5 to 8 million stem cells with RevidaBlue, this is roughly, 5 to 40 billion tissue cells. You do this a few times a day, every day, you understand the enormous metabolic demand that this puts on the body. In order for these cells to multiply well and healthily, they need to have all the vitamins, the minerals, all the machinery to have nice and good effective DNA repair. So that's where AMPM comes in line as a good product to blend with RevidaBlue. And then let's finish with this one, Reveal. Reveal is essentially Luminous for the scalp. So in the same way, as you release stem cells in the bloodstream, you apply Reveal, you will attract stem cells from the blood circulation and then bring them to the skin. If really you have scars and you would like to try what this whole system can do, we've done it in the past. The area where stem cells will go are the areas that are active as an injury, physiologically speaking, for the body. So just take a brush the scar. It will reactivate the scar. Now you put the Luminous on it, take RevidaBlue, and see what this can do for your scars. The product will do nothing. It's your body that does it just by activating this normal process of tissue repair. And I think I've covered everything that I wanted to share with you. Oh, the last one, the last product, MX2O. Again, we talked about it this morning. All these process of tissue rebuilding, tissue repair, tissue regeneration, it brings into play thousands of enzymes. These enzymes will be optimal when they have all the minerals that they need to do their job. So you can add MX2O to the mix. And that's what I wanted to share with you. I think I took a little bit more than the time that I had, right? So.